Hello, everyone. I'm Jerry Wang, and I'm a third-year PhD student in Gabrielle Craman's lab at Boston Children's Hospital. So I did my undergrad in biomedical engineering, and one of the biggest things that I think we all are striving for, and also me personally, is that we want to be called neuroengineers. That's like the big, awesome, cool word that we would like to describe this community, and I think that we're really in the beginnings of being able to finally say, like, oh, we can take intelligence from our collective brains, shrink it down to something the size of a smartphone, and then be able to use it in a meaningful way to transform our environment and to do cool, useful tasks that don't require human labor. So one of the critical things that we kind of have to painstakingly get through before we reach that point is to actually know what's connected with what. And we have a lot of cool research. So for example, here we have nanometer slices using electron microscopy to be able to build synaptic connectivity maps. And of course, we have a lot of people here within this group, like Daniel, and just working on these awesome projects. But there's another aspect of connectivity that I would like to talk about, which is interareal connectivity, and that's not necessarily on the microscale, but between different brain regions. And with the idea of brain regions has evolved so much ever since Brodmann came up with just a handful to now we have over 100. And what I work on is functional connectivity. So how do we link these different brain regions, however they're defined, in a functional way using intracranial electrophysiology recorded from patients with epilepsy. But what I really want to get at today is that there's actually a surprising sort of black spot in the literature of structural connectivity in the interareal world. So the way that we might go about doing this is using post-mortem human brains and being able to track trace using dead tissue. So this is very common in macaques because we can actually inject radioactive tracers while the animal is alive, but it's much more difficult for myelin to take up radioactivity when it's fixed. But I think it should be a rather straightforward challenge that we can tackle today with all of our advanced chemistry and technology that we have to be able to do track tracing within humans. So all the projects that we sort of hear about neuroengineering, like Blue Brain or the IBM 1,000 or 1 million node brain simulation kind of software, we can now finally say with certainty just which brain areas are connected with which. And so far we have the macaque connectivity matrix to work with, but I think there's definitely a lot more work to be done in humans.